Are your people systems ready? Have you opened ChimeraX? Let's start. Last session we have covered about hydrogen bonds, and you have seen that you can also save the information in a separate file. Uh, there were two options, like you can go through the command line and also through your and also through view docx, which is built-in tool for docking analysis in ChimeraX. So we will be using ChimeraX tutorial, so that if you want to repeat it again and again to understand things and also find what are the available options, you can easily find it through internet. So first option is to search for the Google, like ChimeraX binding sites. So like that you will be find the tutorial, this protein ligand binding sites, where you will be getting options how you can use these things for analysis and getting most of it, like how to generate a good quality of figures for your publication or presentation. So I will be referring this for today. So here we can first we will be doing one thing, that we will be using this command in ChimeraX, or like I have opened my ChimeraX and also this web browser, so I can use that command side by side. So here I am clicking this, maybe this will open something, and it should open browser or some option. And if it doesn't work, like I guess it's not working for me, so I have just copied this command here, open help and user tutorial binding sites. I will copy this command. I am copying this command here. I am copying this command here and pasting it in command line, and pressing enter, and it will pop up a window which you can dock side by side here or keep it on one side. So like that this window is open, and I will be following this tutorial for today for finding contacts in classes. So here I will just follow whatever tutorial they have provided. I would suggest you to start, means what I am doing, you should perform the same on your system. This is Chimera browser, and this is Chimera. So if you click these commands here, it will directly uh, control the ChimeraX. So if I click on this open command, it will open a structure over here directly. Okay, like that we were doing some command performing opening PDB structure using open and some PDB ID, and it was opening one structure for us. Similarly, it is doing same thing. So like that you can also create some your own tutorial type of things or built-in commands and list them here or in some file and directly execute them to automate your process. So that will be useful in saving your time and repeating same thing for most of the projects. So like that I have opened it, and here it is giving me some instructions, like we have already discussed about performing uh, means navigating ChimeraX, like model panel logs and other things. And also if we can rotate, zoom in, zoom out, or select molecules, those commands are listed here for details. Means in case you forget something or you want to refer any article, this will be very helpful to you. So like that I am following next command, select clear, and if there is anything is selected, it will unselect all. And similarly, if like we were changing the styles of the or display of the atoms, that will be done we were doing using these molecular display options. Similar thing can be done using commands here. So that is using preset command, and it has changed everything to this representation, means sticks representation. Fine. So earlier we were seeing cartoons and ribbons, but now we are seeing the balls and sticks. Means balls are missing, so it is just a stick representation. And clicking this original means how those atoms are recorded in PDB. So there are few remarks or other settings in PDB structures for representation that you can also reset using preset original option. So like that sometimes you open your protein structure and you see heteroatoms like surfaces, not exactly this ribbon structure or cartoon structure. So that you can do using preset original. Now here other options are there, like I'm using style ball option. So what it does is it will change the representation of a few amino acid to uh, stick and ball. So that will be changed something like this. I'm clicking again this, so this is sticks option, and if I click stick ball, so those atoms will be uh, represented as a ball. So now it has this PDB structure has one ligand named STI. So if this command states that, it will select the STI ligand. So what is happening basically here? Earlier I have already told you that there are selection of different level atoms or groups. So for model selection, they are using has for model and colon for residues. So residues can be number or name. Number means like you in your protein sequence, first residue, or there might be other numbers like 27th residue, 28th residue, or name that can be GLY or GLN something. So that can be used with this combination, and at the rate is used for to specify atom. So this is currently selecting colon st1 and styling it as sphere. uh, so where is that? We will directly click it on it, and the STI residues will be colored like this, sorry, represented like this. So these spherical representation are STL. We can also select them here using select command SEL and colon STI. So those will be selected. Now you can see this green green outline around them, and to make it different, I will use color option in actions to make it red so that you can clearly see it. Okay, so I am just clicking it again, and these are represented as balls, or sorry, spheres. I am going to currently I have selected these residue. So if I click style stick, they will convert it back to stick and stick representation. So here you are seeing few representations, you have seen few representations ball. So the molecules will be represented as ball and stick, like this. There are some atoms. So after this representation, this atom will be become a ball, and the bonds will be represented as stick. So like that, this representation will be changing. And next, if we use stick representation, so if there are atoms something like this, those atoms will, including those atoms and bonds, this will be represented something like this. Okay, and next is your sphere. So with the sphere, if your atoms are here and its van der Waals radii covers some, this is some van der Waals radii from this atom, then for this atom this van der Waals radii, so this will be represented something like this. So within that van der Waals radii, they are bonded, and if they are distance, then they will not be connected, and you can visualize that clearly. And also for protein, you are already seeing cartoon representation, cartoon or ribbon. Now also here it is presenting to change the color of the carbon atoms. I am using changing the color of the carbon atoms of the that ligand. So just clicking that, it will change the color of the ligand, something like this. So currently this tutorial doesn't follow selection, so I will unselect all using this tilde sign and press enter. I am typing command for your visualization. I am using tilde sign. This is called tilde, and other symbol with this is this symbol. This is just left to 1 on your keyboard, and to get this sign you have to press shift plus grave accent button. It is uh, this symbol is called grave accent. Fine. And also you can directly tell the names, like here it is telling name salmon. So list of colors can also be visualized here. So it will be displayed in the log panel, and all the colors available are presented here. Sometimes you might have to change the representation of some selected amino acid or selected atoms which you might want to focus during your presentation, so that can be done here. Now it is telling to select the that ligand. I will again unselect it, and to view complete whole molecule I will be using view, and here again I will start with this tutorial where it is going to perform the H bond and contact analysis. This will by using this command it will be selecting the ligand STI, and view select will show focus the ligand. And if I click hide tilde unselect, so what it will do, it will hide everything else. Now here you are noticing two things, that you are using here you are seeing protein, ligand, and some other head atoms like water. So protein is presented as cartoon or ribbon, and ligand is presented as ballistic representation, and other head atoms are also present with the similar representation. So by typing hide, it is going to hide from this representation, means this atomic representation, not from cartoon or surfaces. So after selecting this ligand, now the here I am performing HIDE tilde and cell. So what it is doing, this selection means selected atoms which we have already selected using STL. So it is selecting the ligand. This command is selecting ligand, and this symbol is negation symbol. Negative means if I say X, so entity referring to that particular command or particular selection will be taken or considered, and if I use this tilde sign, that means other than that selection or that representation, other atoms will be selected or acted upon. So using this, it is hiding all the water molecules. So I will use so not selected. So these other atoms were or these other representations were hidden earlier, and to hide them again I will use hide not selected. So this will hide other than this selected ligand. So I am going to perform the hydrogen bond analysis, and you have already seen that using my tools command, structure analysis, and H bond. Here you have already done that from tools, structure analysis, and H bond. So and also I have told you about the command, and here we are going to use that same H bond command, or you can sort it to HB. I am using that command, and it is going to calculate those commands, and it has those interactions. So it has listed the total hydrogen bonds, and here 23 hydrogen bonds it has found, and those hydrogen bonds are uh, colored in this blue dashes, and these blue dashes can also be customized using options presented here. So also if I am using this cartoon suppress files, what it is going to do, I am going to focus on particular part of the protein, like you are seeing a loop here, right? So this atom, this oxygen atom on the ligand is interacting with this aspartate residue, but you are not seeing its atoms. So if I click on cartoon suppress files, it will expose those residues which are interacting with it. So if I focus here, I can see that this blue color nitrogen is interacting with oxygen in the formation of hydrogen bond. So like that other places, means for other interactions I can clearly see that oxygen of this isoleucine is interacting with the oxygen or oxygen present here. Sorry, it is interacting with the okay, nitrogen present here, and this is oxygen. Actually it was my mistake to color it red, because here heteroatoms were colored, all heteroatoms were colored red. So to revert that thing, I can go here and go to color, and from here by heteroatom, so the oxygens or nitrogens will be colored red and oxygens will be colored sorry, oxygens will be colored red and nitrogens will be colored blue. So here this oxygen is interacting with this nitrogen. Fine. So like that all the hydrogen bonds can be visualized. And also earlier if we add show distance command, so it will be showing all the distances which will be presented here. So I will repeat this command by copying pasting to show the distances. I'm copying and pasting, and this show this true. Actually you need not to type complete keyword, but it is recommended to type complete keyword. So what uh, what is happening here, like if I am telling it to provide option true, I can type t or tr true. So all these work because it knows that at this place anything starting with t is going to be true, so it will consider all those options. And if I start with false and go to till false, it will consider all the options starting with t or f as true, or starting with f as false and t as true. So similarly if I am using h bond command, h bonds command, so either I can type h, yeah, so h may be some other command, and if it conflicts with that, it will show the error. And if I type hb, so there might be no other command, so I can use that, but recommended is that you use h bonds command. So means for shortness you can use follow these shortcuts to save your time. So here I am using cartoon suppress files which I have already used, and now use. So this thing has shown us the uh, hydrogen bond analysis, and here we can see the distance of the hydrogen bond. Fine. So and to get the more information about hydrogen bonds, I will go to this H bond, and if I click on it, it will take me to this H bond tools here, and more information can be visualized here. uh, H bond commands, so I will go to that as well. This is showing H bond commands. Actually we have already performed H bond here, so if I directly click on H bonds, it will tell me all the information about H bond command. There are two things, like one you will see as command and another is as tool means if you are performing anything through these options, that is going to be referred as tool. And if you are using you are directly instructing it to perform something from this command line, it is referring it as a command. So next so I guess this hydrogen bond analysis is complete, understood to you all. Were you able to perform these things? Are you all of you able to perform this much?
Yes, sir.
Or if you tell me I can wait, means it will be done in 4 to 5 clicks only. Or if you tell me I can share this link as well on in the group, should I share?
Sir, I am trying the last one. Hello.
Yeah, you will not get any error here. Is there any problem? Okay, I will wait, Madhurika, and you can just carry on. Don't worry.
Okay, sir.
So in this part, I have just completed the hydrogen bond command, And other options like showing distances or naming these atoms, we can do some additional work. Like our ligands are already selected, so just to display the name of these nearby residues, we can use this um, label and add display, so it will display the names of all the residue nearby. So like that we can visualize that as well. So the it will just display residues nearby the selection. Currently my ligand is already selected, and this command is going to display the residue name as well. I'm typing the command for you. I'm typing here. You can just label and use added display. All this information is already available on the ChimeraX or other places, so you can just utilize that. Subra, Divyanshu, are you performing same thing?
Yes, sir.
So, sir, actually, I'm traveling right now, so
Okay,
I'm
okay.
just.
Okay, no problem. I will upload this video, and I would suggest you to practice this. It will take just 5 to 10 minutes. You have just to click on the these commands. It will be easily done. Okay, no problem. Madhulika, just let me know when you are done. Okay.
Yes, sir, I'm done.
Oh, okay. I thought you are performing the same. That's why I was waiting.
No, sir. Oh, okay.
Okay, so till now you were able to visualize the hydrogen bonds. Like from here, if I am interested in binding of this ligand with some selected residues, I can I can focus on that. Like here, ILE 360 and other residues are nearby. So to select them, those residues near the hydrogen bond, I can also use option select true to select them. So those residues which are nearby will also be selected. So it has selected those residue, and I can color them using action color and other color. I am coloring red so that you can easily visualize them. Fine. So these residues which are colored red are participating or interacting with this ligand here, and next is contacts. So for the contacts, what I am going to do, I am going to close this structure, close this structure here, and I will reopen it and follow the commands again. So for that I will just refollow those commands, like recoloring the ligand using color. So the ligand is colored like this, and hide unwanted things or unselected things. So here I am selecting STI, hide unwanted things like water or other things. So those are hidden here. And now I'm going to move to, I'm moving to contacts. So for the contacts, it is this is the command, and I'm going to use this command here as well. So it has shown me those contacts, and it has not showing it is not showing bonds, because make pseudo bond was false. So I am going to remove this particular option from here, make pseudo bonds false, or I can make it T for true, or completely remove this part. So these green dashes are showing that these residues are interacting, but it is very cluttery, because if any residues is nearby, so multiple atoms are interacting with the nearby residues. And similarly, to display the interacting residue names, I can use label at that at this at the rate display option to display the names of the participating residue. So those will be listed something like this, and here I can hide solvent. So this is hiding the all the water molecules or solvent molecules, and here selection clear. So like that means hydrogen bonds and contacts can be visualized. Madhulika, are you able to perform this much? Means, or anyone else is
Yes, sir.
following me here? Okay, so I will move to next option. Also, I have already said that you can visualize sequence, but from this tutorial I will just do a few things, like it is I am going to see the sequence of chain A. I am doing something selected, but you can use, I would suggest you to practice these things and read the instructions here. So it has shown me the residues, it is showing me the residue sequences of chain A, and if I select the contacts, then it will also highlight those contacts here. So to select the contacts, I will again use the same command, and also use select true option, select T. So it will select them, and here those selections will be highlighted dark green, these green colors. So these green colors are selected here, means that ligand is interacting at with these green color residues. And also you know that these yellow and light blue representations are for uh, helix and loops. So you can also perform your analysis or interpretation based on that. So here we have completed ligand contacts and hydrogen bonds. So I'm just limiting to this hydrogen bonds and contacts. More information can be found over the internet, means if you want to go in depth, I would suggest to go through that. Contacts and S bonds. So here first tutorial is classes. So here you will be getting the information about contacts and what are the classes, because in the PDB structure there are like overlap, atoms can be overlap within the van der Waals radii, so that are called classes and something. So all that information can be obtained from here, and those default options can also be found here. Means these things are going to vary for your studies. So I am just introducing what are the available options for your understanding. Now since we have already in our results we were all able to cover the hydrogen bonds, contacts, and also the score. So based on that we can select our best uh, best residue. For that I will open my docking, older docking, where I have listed all the hydrogen bonds. So what I have done here, I have just performed the docking some with some receptors and ligand, and then I have used their conformer name, that like that in the vina, means from vina directly. So these are conformer names and these are their conformers scores. And here I am getting contacts, means how many contacts those atoms are making mutually and how many hydrogen bonds are being formed. So I have also listed these contacts, means contacting residues here one by one. So these means for these 65 contacts I have listed all the residues by copying pasting, and you know that you can save those contacts and contacts using a file option, which you can provide using save and file address. So that is going to be saved in your directory and you can use that for to list them out here. So like that I have stored the contacts, sorry, I hide a column with 3. Okay, so and next I have selected these hydrogen bonds. I need to move it. Better I delete this column for clear visualization. So for one hydrogen was formed with this residue, and here ligand was donating, here arginine was used in donation. So like that here in conformer 5 I was able to see that there are four hydrogen bonds were formed. Like it was formed by arginine, means where this was donating, arginine was donating uh, hydrogen, and ligand was also donating hydrogen. So there were four hydrogen bonds were being formed. Similarly for the earth residue there was tyrosine, and it was uh, donating hydrogen to ligand, and also ligand was also donating hydrogen to the tyrosine residue. So like tyrosine or other residues nearby. So like that I was able to log those things. So since here contacts are like how many atoms are nearby, so based on that I am going to select best pose for me. And also since I might be interested in the residue sequence where it should be interacting, so based on that I will be selecting my best pose. So like here if I am using some protein where some other ligand was binding or some other residues were interacting, I will be selecting based on that residue. So for my study I found this pose better, where there were means involvement of tyrosine residue, and there were multiple hydrogen bonds and contacts were also good. So for this protein 2AST I found uh, this conformation better. So what I did, I just deleted other conformation using ChimeraX and saved this protein and residue complex, sorry, protein and ligand complex for further study. And similarly for the second one, my selection was different. So here I selected this third conformer for my study. Means further we perform mostly molecular dynamics study. So this complex was further taken for the molecular dynamics using you can perform using any software like GROMAX or whatever is available with you. So like that I was able to use these docking results for my molecular dynamics study. And if I find that this receptor and ligand reacts means interact properly, and there it shows stability in interaction, I can use this information for further to validate in vitro, or means to prove my hypothesis about some pathway or biological process. So like that, from docking to in vitro, we can create a pipeline or one understanding to complete our proposal, or our some experiment, or find some new drug, or predict the working of some existing drug. So I guess till this much you all were able to understand these things. So here is assignment for you all, that you just select one existing paper which you have selected, or select a new paper, and there you just analyze what docking they have perform that docking, and also analyze the results, and check what were the parameters, or the outcomes they have listed. Like here I have just simply told you to note down the hydrogen bonds and contacts, but they might have added some additional information, like this some particular they might have focused on some particular residue or interaction of particular sequence. So you can also highlight that part as well. So for that study from that study, you have to log the outcomes from there and comment that, and I would suggest you to write it in a formal way, so that like something like a research paper, where you write the results part. So like that you can do that. And also next, one more thing you have to do, that for the understanding of that particular research paper, what were the problems or difficulties you were facing, I would suggest you to share those things as well as a comment. And also since you know few things, so also few things that you have some additional knowledge from here, or they have performed study earlier, means in older time. So what will be your suggestions, or based on your understanding, to improve their study? So you can add, I would suggest you to add that as well. And next, your questions. Next time I will be covering these multiple complexes, means how you can perform docking for multiple complexes, or perform docking using other software or servers, that I will be covering tomorrow. Just I will share the resources, like other web servers or something else, so that you have other options as well with you. Like sometimes we are not able to use our local system for the docking purpose, or there might be multiple complexes which might not be suitable to be performed on local system, means on our laptops or desktop. So we might have to use servers. So I will share those options. Like Vasu is not able to uh, perform all the things in his system, so he can use other online servers. I will share those things. And is there any question from this session today? Divyanshu, you had question in the group, you were asking.
Yes, sir. I had a question. Sir,
Yes.
how will we get like more negative energies? I saw in your results you had -11 affinity. I'm not able to get anything above -6.
See, I would suggest to one thing, that So you are already understanding that if two atoms are interacting with each other, then there will be some interaction, and based on that interaction, means type of interaction, there will be some free energy type of thing, negative energy
Sir, yes.
for the stability of the system. So for that there will be some energy E1, and for other system there will be some other type of energy E2. So this energy sometimes could be more, E1 could be more than E2, and or E1 could be less than E2, or they might be equal. So this all these things depend on the environmental factors, like if you are putting your lipid molecule in water, so there that might not be showing stability, or if it gets proper folding, means aggregation, then it might show some stability. So like that, environment also affects the stability of the system. And next, like for the protein, if protein folds stop spontaneously, and for protein folding, you might have also observed that protein the polar residues orient outer side on outer surfaces, and those hydrophobic residues try to uh, tend to orient towards inner side, where all the hydrophobic residues are present. So that is basically to minimize the to minimize the energy of the system and increase the stability of the system. So increase the stability of the complex participating in those interactions. So based on that, you will be getting optimum energy. So so there is no option to increase or decrease the energy. Next thing is that if you are thinking that a protein, some protein P1 is interacting with ligand 1, and if you want this ligand having some weak interaction with this P1, so what you can do, you can modify the ligand, like it has some atoms, you can replace some particular atom, and and add some other atom, and then if you perform docking, so it might change the interaction energy, means it will make the system more stable. Basically what this is how we find new drugs. Like in my lab, there are like study on ppr gamma or delta. So for those, or we can also ppr gamma or delta proteins. So for that we are having some ligands. uh, Let us take example ligand 1. So this is the base ligand, and based on this there are multiple modifications we have performed and also synthesized in our lab. So what we saw that this ligand is having some was giving some energy like -5, but the other modified ligand L1A, L12A, here I'm naming it as sorry, I'm naming it as L1 modified 1. So there we change some other atom, means replaced some functional group. Like you have already seen the chemical reactions or studied chemical reactions, where people are replacing some existing atoms or a group of atoms to with other reacting group. So like that, here we modified this base molecule with the other molecules L1L, and similarly L1 test and 3. So we made multiple 8 to 10 modifications, but in the docking results few molecules were giving lower energy, like let us take example, this molecule was giving lower energy than this L1, but these were giving better energy than this one. So this modification of the atoms within the existing ligand was helpful for us to give better binding energies. And based on that, there were like a few experiments were also performed, like different assays in vitro, those also suggested that this activity of this molecule is better, means these new molecules or synthesized molecules are better. So like that means we vary to find the favorable results, we do these modifications, and like that drug discovery works. So if you are getting lower energy, don't worry about that. And next thing is that if you are performing docking, I would suggest you to check for if you have added hydrogen bonds, because during analysis or during performing docking or dynamics, hydrogen bonds also matter, or there might be some issues like in the while you are performing the analysis part. So in the analysis part, there might be other heteroatoms like water molecules or others, so that might be hindering these interactions. So I would suggest to try removing that. Means they might not always hinder that, they might also help that those interactions, but it depends if drug is giving proper good results or not. So I would suggest you to try with a water molecule and after removing that as well, because we all know that molecule can directly interact in a better way, and also if water molecule is present, so that can also assist in binding of ligand with the protein. So that can also be the case, uh, but you cannot means change the binding affinity except these parameters or environment of the receptor and ligand molecules. So do you have an other question, or
What is the minimum threshold that we can consider is a good for binding?
there is no threshold, because if you see protein that has some n number of atoms, and there is another protein which has n more than n number of atoms, like some protein has 200 residues, fine, and another protein has some 500 residues. So with this the result is going to be different, and with the if ligand has some different number of atoms, like uh, 7, and other ligand, what happened, suppose protein has 100 residues, and another protein has 200 residues, and ligand has some 10 atoms, and another ligand has 17 or 18 atoms. So if we check that this system is going to be stable with minimum energy, like some let us take example of minus 4, because number of atoms are less here, so outcome of the energy will be lower, because that is not the relative, means that the energy you are getting is based on the number of atoms, means interacting species. But if here the result will be varying, and here it might be more than minus 4, like minus 7 something. And but for similar this system, this could be more than minus 9, let us take example, it is 9, -9. And for this is going to be more, like it might be some -12 or something. So like that based on the interactions or nature of the molecule, these energies are going to be varying. So there is no set criteria that you have to uh, use that particular cutoff. And if someone is using that cutoff, that means that is like they are only studying based on some particular protein and receptor, particular protein receptor or particular ligand. So based on that they are setting up some criteria. So that criteria, like let us take example, I have some these 20 ligands maybe, let us take it as example. So uh, here if I am getting some variable energies, so I can set criteria to filter this, but not as a criteria to tell that this is not a good binding or bad binding. I can like if I am getting some 50 ligands, and I want to use get top 5 results. So for that I will be setting cutoff criteria, not for to tell that these interactions are good or bad. So that is not going to be helpful. Means I would suggest not to go for that.
Okay, sir. Thank you.
Was I clear?
Yes, sir. Yes, sir.
Any other question from anyone?
No, sir.
Okay, so I would suggest to suggest you to perform uh, to for do the assignment because it will be helpful to you all. I will share this in the group. You have just to replicate the existing research papers. Any other question? Any other person? Any random query will also work. Means if you have any type of question, you feel free to ask. Don't think that because I also don't know. I just learned it like a few months back, 5 to 6 months ago, and I am sharing those things with you. Okay, so I will terminate the session, and if you have any query, feel free to ask in the group as well. Okay.
Okay, sir.
Okay, sir.
Good night, everyone.
Good night, sir.